Okay, this is our final video, and it's really just to show you examples of how cool inhibitors can be and how highly related they are to human health. Let's remember, we have talked about three general classes of inhibitors. There's the two types of inhibitors that are reversible, competitive and non-competitive. And these are distinguished by the fact that one binds in the active site, competitive, and the other binds in a different site and is non-competitive. Lastly, we had the irreversible covalent inhibitors. We haven't really talked about those yet, but they bind covalently and therefore permanently. Let's push way to the very end of the chapter, and I want to go through some example inhibitors to share with you of how interesting and important they can be. I want to talk about pandemic, not COVID pandemic, but rather influenza pandemic. And the flu, like H1N1, has caused some terrible pandemics in our history. And the most recent H1N1 flu outbreak that went really heavy was in 2009. Now, the N of H1N1 stands for neuraminidase. That's an enzyme that's part of the flu virus, and its job is to chop a sugar off of cells in our respiratory tract. So here we have a cell. Let's just say this is a cell, and it's got this sugar. That's what we're talking about next, by the way. Next chapter is sugars. And what the virus does is it chops that sugar off and separates that sugar from the cell as part of the life cycle. And if the neuraminidase enzyme can't do that, then the flu virus can't release and go on and to infect other people. So people have thought about this for years about could they inhibit this enzyme? Could they design inhibitors that might prevent the flu from spreading by inhibiting this enzyme? There's many different enzyme inhibitors that have been proposed, and one of them is called zanamivir, and it's very highly related to something called Tamiflu. You may see this from time to time. I don't think this is actually Tamiflu, but it's very similar to it. And Tamiflu is something that you can take when you get early stages of symptoms of flu. It doesn't really help that much, to be honest with you, but it does a little bit. And notice that the particular structure here looks very similar to the structure of this sugar. In fact, here's the sugar over here, and here's the inhibitor. Notice how similar they are. They're not identical. You can see there's a double bond here and a single bond here. There's no alcohol here. There's an alcohol here. There's a big guanidium group down here, missing over here. This thing is a competitive inhibitor. If the substrate fits in the active site, this is like a very similar key that's going to fit into the very similar lock. And it goes in there, prevents this from chopping that sugar off, and therefore slows the flu virus down. Here is another inhibitor of neuraminidase. It's called dihydroxyxanthone, and it's an inhibitor of this enzyme. It's not a very good one, and it's not a terribly good lead for a drug, but nonetheless, I like to include it because it's an example of a non-competitive inhibitor. Notice that this inhibitor is a flat aromatic ring. It doesn't look anything like the substrate that we see above, and that's very typical of a non-competitive inhibitor. It's got to bind somewhere else, and therefore it's going to bind in a different fashion. Now lastly, I want to show you one more example. This is a example of a drug that was under development. I don't think it ever became a drug, but this is called DFSA. And some scientists thought that this might make a pretty good inhibitor. Notice it looks an awful lot like the substrate at the top of the page, but it's got a couple of fluorines on it. That's a little unusual. And I can tell you what happens is this drug is going to fit, here's the drug, it fits in the active site, and the active site would be sort of like this. And the active site has a couple of catalytic residues that is residues that are involved with the chemistry. It's got a aspartate right there, and it's got a tyrosine right there, our old friends, the amino acids. And these actually undergo some chemistry. There's some acid-base chemistry. There's a nucleophilic attack. This fluorine is a good leaving group. And what you end up with is a covalent bond right there between the enzyme and the inhibitor. And once it forms that covalent bond, it's permanent. It's stuck. And it's in the active site. After all, it looks like the substrate. So it gets in there and mucks up the work. It's kind of like saying, well, a competitive inhibitor would be a key that you can put in to a lock and it blocks the binding of another key. This is like a key that fits in the lock and then you break it off and it's permanent in there. Or it's like putting glue in a lock so you can't get the regular key in there ever. This is a very effective type of drug, and that's because once you get one molecule, it destroys one active site and one enzyme permanently, and that enzyme can never do its job again. So, like I said earlier, there's lots of interesting drugs like aspirin and penicillin and a lot of others that might be very effective drugs, and I wanted to mention that as an example of covalent permanent inhibitors. That's the end of this video and the end of our chapter 2.